Hello, everyone. My name is Alessandro Busetti, and I am the lead field application scientist for the international business team here at Metabolon. And today I'm going to present some research on infectious diseases, and in particular on malaria, showing how metabolomics approaches can be used to characterize the blood stage parasite development, conduct biomarker discovery, and identify novel therapeutic targets. So, in particular, the study that I will be talking to you about today was published in the Malaria Journal in February 2020, and the title is Metabolic Alterations in the Erythrocyte During Blood Stage Development of the Malaria Parasite. Now let's begin with a bit of background. Malaria is a mosquito-borne infectious disease affecting humans and other animals, which is caused by a single-celled microorganism, in specific a protozoa. Protozoa comes from the ancient Greek protos, meaning first, and zoion, meaning animals, so first animals. And these protozoa are of the Plasmodium genus. There are 5 species which regularly infect humans: Plasmodium vivax, falciparum, Malaria, ovale, and knowlesi. However, Plasmodium falciparum is by far the most lethal in humans, resulting in hundreds of thousands of deaths per year. In fact, according to the World Health Organization, in 2018 alone, there were about 228 million cases of malaria worldwide and approximately 405,000 deaths. Now, a number of drugs have been developed to treat Plasmodium infection. However, the parasites have evolved resistance to many of these drugs. As you can see on the image on the right, Plasmodium falciparum has a complex life cycle. In humans, the parasites grow and multiply first in the liver cells and then in the red blood cells. And in the blood, successive broods of parasites grow inside the red blood cells and destroy them, releasing daughter parasites called merozoites that continue the cycle by invading other red blood cells. The blood stage parasites are those that cause the symptoms of malaria. When certain forms of blood stage parasites are ingested during blood feeding by a female Anopheles mosquito, they mate in the gut of the mosquito and begin a cycle of growth and multiplication in the mosquito. After about 10 to 18 days, a form of the parasite called a sporozoite migrates to the mosquito's salivary glands, and when the Anopheles mosquito finally takes another blood meal on another human, it injects anticoagulant saliva, and together with that, it also injects the sporozoites, which migrate to the liver, thereby beginning a new infectious cycle. During the symptomatic stage of malaria, Plasmodium falciparum resides in human blood cells as it multiplies asexually during the 48-hour intra-erythrocytic developmental cycle, also known as IDC. The human erythrocyte is also the main conduit for providing Plasmodium with essential nutrients during its development in the IDC. Now, while the interactions of the parasite with its host, so the human erythrocyte, have been studied for well over a century, much remains to be discovered and clarified. For example, although parasite-infected erythrocytes rapidly sequester arginine from the culture medium under in vitro culture conditions, the relevance of this to the parasite development is still unclear. This study provides an example of how high-resolution metabolomic methods have been employed to improve the understanding of host-parasite interactions with the aim of ultimately identifying novel treatments and diagnostic strategies. Now, before showing you and discussing the results, let's go through what the researchers of this study did. So the researchers obtained Plasmodium falciparum NF54 parasites and propagated them in human erythrocytes, which they obtained through phlebotomy. They kept them at 2% hematocrit in gassed flasks. They were obviously maintained in a specific medium. And then they had to synchronize the parasites. In fact, as I said previously, Plasmodium falciparum goes through several life stages. And to generate synchronized parasites, the cultures were passed through magnetically activated cell sorting columns and purified every 44 to 48 hours for 4 days before the initiation of the experiment. The researchers used blood smears and light microscopy to monitor parasitemia and synchronicity, and additionally, they also conducted PCR to confirm the absence of mycoplasma. Finally, the cells were harvested from the 4 test flasks and the 4 control flasks, so the uninfected cultures, and flash frozen in an ethanol dry ice bath and stored at -80 for subsequent metabolomic analysis. This procedure was repeated at the following times, so during the IDC, so 0, 8, 16, 24, 32, and 40 hours. So in this slide, you can see the results of the global metabolomic profiling of uninfected red blood cells and infected red blood cell cultures. Now, using Metabolon's untargeted global metabolomics platform, 501 metabolites present in either uninfected red blood cells or infected red blood cell cultures were quantified. Of these, 481 were present in both cultures, and 20 were unique to the infected red blood cell cultures. In figure A, so in the top left, you can see the individual log base 2 fold change values for all identified metabolites as measured for each replicate at each time point. Compared to the infected red blood cell cultures, which showed increases in fold changes values late in the IDC, so at 32 to 40 hours, the uninfected red blood cell cultures showed fewer metabolite changes over time. Now, to determine the overall characteristics and time-dependent alterations of the metabolite data, principal component analysis was performed on the fold change data. And in figure B, so on the bottom left, you can clearly see a separation of the data for the 2 culture systems over time, with the 4 replicate data points for each time point allowing visualization and assessment of the data spread. The untreated red blood cell cultures underwent metabolic changes over time, as shown by separation along the second principal component between the 0 and 8 hour and also the 16 and 40 hour time points. At the last data point, 40 hours, there was less separation between the uninfected red blood cells and the infected red blood cell cultures. That is, the metabolic profile of the 2 cultures corresponding to the late stage schizogony of the parasite began to resemble that of the uninfected cultures at 16 to 40 hours. Now, on the right, very important, to identify the underlying groups of metabolites that drive the changes in metabolite profiles, hierarchical clustering of all 501 metabolites was performed, and metabolites that behave differently in the 2 culture systems were characterized. In this figure, you can see that most metabolites can be grouped into 5 distinct clusters based on their wide-ranging changes over time in response to parasite infection. So, cluster 1 consists mainly of metabolites that were consistently lower in uninfected red blood cell cultures than in infected red blood cell cultures, and higher in infected red blood cell cultures than in uninfected red blood cell cultures across all time points. Cluster 3 exhibited a trend opposite to cluster 1, whereas infected red blood cell cultures metabolites in clusters 4 and 5 showed marked time-dependent changes associated with IDC progression. Cluster 2 contained the largest number of metabolites whose fold change values were mainly of smaller magnitude and less dependent on time compared to the other clusters. Now, cluster enrichment analysis was used to determine whether specific metabolite classes could be associated with these clusters. Clusters 1 and 5 were enriched in nucleotide metabolites, clusters 2 and 3 in lipid metabolites, and cluster 4 in carbohydrate metabolites. This is commensurate with the known global metabolic features associated with parasite-infected erythrocyte cultures during the IDC. So, in fact, both time-dependent and time-independent production of nucleotide metabolites in clusters 5 and 1 respectively, consumption of carbohydrate metabolites in cluster 4, and an increased depletion of lipids needed to create membranes for a parasite progeny in cluster 3. So this slide describes the quantification of consistent large metabolite changes between the infected and uninfected cultures, an approach that can potentially identify circulating metabolite biomarkers indicative of malaria infection. And 93 metabolites increased or decreased twofold in average abundance in infected red blood cell cultures relative to the uninfected cultures. Now, the table on the left shows the 15 metabolites that increased most in abundance, with fold changes ranging from 4 to 64, which is pipecolate. These belong to clusters 1 and 5 and correspond to metabolites that either consistently increased in infected red blood cell cultures, so cluster 1, or which showed a clear time-dependent increase in abundance at later time points, so cluster 5. Now, the table on the right shows the 15 metabolites that decreased the most in average abundance, with fold changes ranging from 3 to 33, in particular fructose 6-phosphate. Now, these metabolites, which all belong to cluster 4, were representative of a set of metabolites that consistently decreased in abundance in a stage-dependent manner with IDC progression. The largest increase in metabolite abundance occurred for pipecolate, as I said, which is a product of lysine catabolism. The increase in pipecolate was associated with the schizon stage, so 32 to 40 hours of the infected red blood cell cultures. Furthermore, pipecolate, which accumulates in the plasma and urine of patients with severe malaria, has been suggested as a candidate clinical biomarker of the disease. Pipecolate is strongly associated with parasite-infected cultures and may be necessary for IDC progression. It is also associated with increased inflammation, oxidative stress, and epilepsy. The largest reduction in metabolite abundance occurred for fructose 6-phosphate, a product of glycolysis breakdown, reflecting the high glucose consumption capacity of parasite-infected erythrocytes. Within infected erythrocytes, most of the glucose, let's say 60 to 70%, is in fact incompletely oxidized to lactate and then excreted. For all metabolites present in both uninfected red blood cells and infected red blood cell cultures, the observed differences in abundance represent both a red blood cell response to infection and the parasite component, providing a direct measurement of the intraerythrocytic environment. And I forgot to mention that the data in the previous slide also points to significant changes in a very specific lysophospholipid, which has never been reported before. So that's particularly interesting. Now, in this slide, you can see the global and temporal metabolic activity increase during the intraerythrocytic developmental cycle. The main thing to notice is that the infected erythrocyte cultures exhibit continuous and distinct metabolic activity. And in fact, the figure A, the one on the top left, shows the fold change in the abundance of the top 93 metabolites at the 6 measured time points. The magnitude of these fold changes increased from early to later time points. And in figure B, you can see the number of metabolites changing by at least twofold in abundance at each time point increased from 42 at time 0 to 87 at time 40, suggesting a parasite-induced increase in metabolic activity following infection and a growing divergence of activity between uninfected red blood cells and infected red blood cell cultures over the course of the IDC. Importantly, although the infected red blood cell cultures were less active in the early stages than in the later stages, they were not metabolically inactive. And in figure C, you can see the detailed fold change in average abundance of the metabolites from clusters 1 to 5. So next, the researchers looked at the influence of metabolite alterations on metabolic pathways. and to identify and quantify the changes among these metabolites associated with parasite development during the intra-erythrocytic developmental cycle, pathway enrichment analysis was performed on all metabolomic data, which is what is shown in the figure. Metabolites of the amino acid class, such as those produced by arginine, proline, aspartate, and glutathione metabolism, showed significant enrichment at intermediate and late time points, consistent with their canonical role in protein synthesis and oxidative stress. This enrichment was associated with hemoglobin degradation, which provides a major source of amino acids for Plasmodium parasites. In contrast, lysine degradation was the only amino acid pathway significantly enriched at all time points, suggesting that it continuously functions throughout the entire intra-erythrocytic developmental cycle. None of the carbohydrate or energy metabolism pathways were significantly enriched. However, the pyruvate and tricarboxylic acid cycle pathways both showed enrichment at intermediate time points. This enrichment was associated with L-malate, which increased by 2.5-fold. Neither Plasmodium falciparum parasites nor red blood cells have a fully functional tricarboxylic acid cycle, which suggests that the parasites produces malate for incorporation into purine nucleotides via oxalacetate and aspartate. Among nucleotide metabolism pathways, purine showed enrichment 6-fold, as did pyrimidine, a metabolite which is synthesized de novo by the Plasmodium falciparum. Within the lipid class, phospholipid metabolism was significantly enriched, more than 10-fold at all examined time points. This enrichment was associated with metabolites involved in the synthesis of phosphatidylcholine and phosphatidyl-ethanolamine, which together constitute about 75 to 80% of phospholipids in the parasites. The enrichment in linoleic acid metabolism was associated with an increased abundance of polyunsaturated fatty acids. Now, all of these metabolites showed increased abundance in the infected red blood cell cultures relative to the uninfected red blood cell cultures. Overall, the enrichment in each metabolite subclass and associated metabolites showed characteristics of typical parasite development, where amino acids are used for protein synthesis, nucleotides are synthesized for DNA production, and lipids are synthesized for the development of lipid membranes. So next, the researchers looked at the lipid metabolism in specific of infected and uninfected cultures. And in figure A, so the top left, you can see the fold change in abundance of phospholipids, where GPGs and DGs, so diacylglycerols, show the largest increase. LPGs in Plasmodium falciparum may have some additional functions such as cell proliferation, migration, cell adhesion, or calcium signaling, which have been identified in mammalian cells. In figure 5b, so on the bottom left, you can see the distribution of fatty acids with different carbon chain lengths in infected red blood cell cultures relative to the uninfected cultures. Now, apart from the increase seen in a 5-carbon fatty acid, 5 polyunsaturated fatty acid metabolites increased 1.5-fold in the infected red blood cell cultures relative to the uninfected cultures. These PUFAs cannot be synthesized in mature erythrocytes as they lack an essential enzyme necessary for fatty acid biosynthesis. In contrast, Plasmodium falciparum should be able to synthesize PUFAs because it possesses the necessary elongases and desaturases. Now, PUFAs are precursors of eicosanoids, which have immunosuppressive roles. So Plasmodium falciparum under in vitro condition can produce eicosanoids when supplemented with a PUFA. And eicosanoids also mediate fever and inflammation and have numerous other functions. This data shows that the parasites under in vitro conditions can produce precursors that mediate inflammatory and immunosuppressive responses in an infected human host. So then the researchers decided to study the metabolic changes that characterize parasite development. So this section considers the metabolic changes in infected red blood cells and uninfected red blood cell cultures that capture the development of the parasite during the intra-erythrocytic developmental cycle. And figure A shows three key metabolites of glucose metabolism in uninfected red blood cells and infected red blood cell cultures. And as you can see, glucose remains stable in the uninfected cultures, whereas it decreased roughly linearly in the infected cultures, ultimately becoming exhausted at the end of the intra-erythrocytic developmental cycle. Concurrently with the 8-fold decrease of glucose in infected red blood cell cultures at the 48-hour time point relative to the 0-hour time point, there was a 5-fold increase in lactate. And these observations suggest that approximately 60% of the consumed glucose is oxidized to lactate in the infected red blood cell cultures. Phosphoenol pyruvate, so PEP, a glycolysis intermediate, showed higher abundance in the infected red blood cell cultures relative to the uninfected red blood cell cultures early in the intraerythrocytic developmental cycle, so 0 to 16 hours, but only half the abundance in the uninfected red blood cell cultures later in the IDC, so 24 to 40 hours. Plasmodium falciparum converts PEP into products such as pyruvate and oxaloacetate, which are important metabolites for pathways including the TCA cycle. The increased utilization of PEP at the later time points suggests an increased use of the TCA cycle reactions late in the intraerythrocytic developmental cycle. Now, if we remember, there was a significant enrichment in phospholipid metabolites associated with the synthesis of phosphatidylcholine and phosphatidylethanolamine, which comprise up to 50% and 45% respectively of the total phospholipid content in purified parasites. Now, in figure B, you can see the temporal variation in phosphocholine and phosphoethanolamine, which are precursors of phosphatidylcholine and phosphatidylethanolamine, respectively. So both metabolites exhibited the highest abundance in infected red blood cell cultures towards the end of the intraerythrocytic developmental cycle, which is commensurate with the increased demand for membrane lipids in the developing merozoites. Now, the parasite also needs to synthesize purine and pyrimidine nucleotides for DNA synthesis. And it does this by salvaging purine nucleotides from the host erythrocyte and synthesizing pyrimidine nucleotides de novo. And in figure C, you can see the temporal profiles of 3 pyrimidine metabolites, which are synthesized in the first, second, and third steps of pyrimidine metabolism, respectively. All 3 metabolites from the infected red blood cell cultures showed a temporal pattern consistent with Plasmodium DNA synthesis. In contrast, these metabolites were stable in the uninfected cultures because pyrimidine metabolites only exist in small concentrations in human erythrocytes. So taken together, the pattern of metabolic changes in the infected red blood cells shown in this figure reflects some processes necessary for parasite development during the IDC, so during the intraerythrocytic developmental cycle. And finally, what you see on this slide is the metabolites uniquely associated with parasite infection, and in particular the infection stage specific metabolite alterations. Obviously, to identify infection specific metabolite alterations at the early, so 0 to 8 hours, intermediate, so 16 to 24 hours, and late, so 32 to 40-hour stages of the intraerythrocytic developmental cycle, the variation in abundance levels was examined as a function of the infection status of culture and time. Two-way ANOVA conducted for each of the 501 metabolites at each stage with time points 0 and 8, 16 and 24, or 13 and 40, and infectious status infected and uninfected as the between subject factors revealed that 42, 107, and 36 metabolites showed an infection-specific change at the early, intermediate, and late stages, respectively. The figure shows the percentages of these significantly altered metabolites that were associated with one of the major metabolic pathways, or a pathway category designated other for metabolites that do not belong to any of the major pathways. And overall, there was a relative shift from lipid to amino acid metabolism during the progression of the intraerythrocytic developmental cycle. At the earliest time point, so 0 to 8 hours, significant changes occurred more frequently for metabolites belonging to the lipid metabolism pathways. At the intermediate time points, the absolute number and relative fraction of lipids decreased, and the relative contribution of amino acids increased. The decrease in lipid metabolism was also associated with a change from sphingolipids to lysophospholipid metabolism, which included metabolites that appeared uniquely in infected red blood cell cultures, such as 1-palmitoyl GPG and 1-stearoyl GPG. Both of these, which belong to the LPG class, increased more than threefold in abundance during the intraerythrocytic developmental cycle, suggesting that they are functionally important for parasite metabolism. In mammalian cells, LPGs are known to increase intracellular calcium, although the receptor that mediates this increase is still not known. And late in the IDC, so in the intraerythrocytic developmental cycle, so 32 to 40 hours, the fraction of lipid metabolites remained similar to that at 16 to 24 hours, although it now consisted of more phospholipids. Now, the ANOVA analysis overall suggests a dynamic progression of metabolic activity in the infected red blood cells commensurate with highly regulated processes of parasite development and the shifting demands placed on parasite metabolism. This is reflected in changes in lipid metabolism that are apparently correlated with parasite stage, a shifting emphasis from sphingolipids and lysophospholipids involved in cell signaling to phospholipid metabolism related to the de novo synthesis and the accumulation of infection-specific membrane components. And to conclude, I will try and summarize some of the main study insights and give you a list of some of the conclusions. So a new phospholipid class of LPG metabolites present only in parasite-infected cultures was identified. And although these metabolites modulate calcium signaling in mammalian cells, their functional roles in Plasmodium falciparum biology is still unknown. The relative abundance of these polyunsaturated fats in the infected red blood cells increased. These metabolites are precursors for synthesizing eicosanoid signaling molecules, which when released in the host blood plasma can modulate fever and inflammation under in vivo conditions. They're also known to promote gametogenesis and thus may play a role during the blood stage development of Plasmodium falciparum. The improved characterization of lipid metabolites reveals highly dynamic usage of phospholipids during the intraerythrocytic developmental cycle. Specifically, the results suggest that sphingolipid and lysophospholipid metabolites are significantly utilized at early and intermediate stages of the IDC, whereas phospholipid metabolites dominate during the late stages. Overall, the metabolomic data presented here provides a great opportunity for targeted discovery of metabolic functions and biology in Plasmodium falciparum. And to conclude, I would like to thank you all for your attention. And obviously, if you want to know more about how we can assist you in your daily research, please contact us at Metabolon. Thank you very much again.